pipettes to pH probes to parafilm, there is a lot of stuff in a biochemistry lab, and so here is what's what when it comes to wet work. A guide to some common biochemistry and molecular biology lab equipment, so you're more familiar with your surroundings and your lab can become like your second home.
Let's start with pipettes, and there's a lot to pick from. If someone's talking about a pipette in biochemistry, they're typically talking about a micropipette. Micropipettes come in a variety of sizes, ranging from being used for about 1 to 2 microliters to being used for a milliliter. And so a microliter is going to be a millionth of a liter or a thousandth of a milliliter, and it's a common unit that you use in biochemistry because we're often dealing with really, really small volumes. These are micropipetters, and so you can see that you have different ones for these different size ranges, and they're each going to have different size tips. Now, you want to use the size that is the smallest that will still be able to accommodate the volume that you want. So, for example, with the one that can do 1,000, we call this a P1000. The one that can do 200, we call it a P200. 20 microliters, we call it a P20, 10, a P10, and 2, a P2. Now, it'll tell you the range that these can do. So, for example, a P200 can go from 20 to 200 microliters, and a P1000, this one can go from 100 to 1,000 microliters. Say you wanted to pipette 150 microliters. You could technically do it with either of these, but you're going to get the most accurate volume if you use the one that can do a smaller amount. It goes to a smaller amount. You're going to get better accuracy and precision. So you want to go with the P200 in that case. Now, if you forget which one's which, you can always look at the top end. They'll have the numbers listed probably. These also say XLS. This is basically just like this brand thing, and they use these extra long tips and things like this. Speaking of tips, there's a lot of different tips to choose from. And so here's just an example where you can see these are both tips for P1000. Now they have slightly different lengths, but the real difference is that this one has a filter. And so we have these filter tip pipettes. These are really useful for if you want to protect your sample from what was in the pipette already, like on the end part, or if you want to protect your pipette from what's in your sample. So, for example, you might want to protect your sample if you're working with RNA or something really delicate, something you want to keep sterile. And you might want to protect your pipette if you're working with radioactivity or something, and you don't want, when you're pipetting, for things to splash up. Much more on pipetting in another post, but we're just talking mainly about equipment here. So in terms of these tips, in addition to having filter and non-filter, you can have them come in these racks that you kind of refill the boxes from. You can have them come in these individually packed sterile boxes. You can have them just come in bulk in bags where then you stick them in the racks. The ones that are non-sterile, you can typically, we often autoclave them to sterilize them. We'll talk more about autoclaves later. With the filter tips, though, you're not going to want to autoclave those, but those typically come sterilized already often.
There are also these like long skinny tips, these gel loading tips. These are really great for getting into the wells of gels without like making the wells like mad.
So that's the basics of these micropipettes. The second type of pipette you will commonly use, we'll talk more about some like variations of these micropipettes because this was just a single channel. But the other common type of pipette you'll often use is a serological pipette. And with these, you use this like pipette-man or pipet-aid typically. This is going to be an electronic kind of sucker, which is going to basically suck sample up through these long plastic tubes. There are different, basically this pipette-man replaces those like bulbs, which replace like mouth pipetting, where people actually like literally sucked it up with their mouth, which is not a good idea. But these basically use electronically to do this. They have some that are kind of like a cylinder where you use a scroll wheel to pull things up and down. But these pipette men are really, really helpful. You can see that they typically have a filter plug in them so that you don't accidentally suck things up too far. And then there are different gradations on these different pipettes. And you can see that they come in a variety of sizes. So we have like a 5 milliliter, a 10 milliliter, a 25 milliliter, a 50 milliliter. And just like we were talking about with the pipette tips, we're going to want to choose the smaller size that can do the job. And in this case, it's because the lines are going to be closer together. We're going to be able to measure more accurately, because you can only measure basically as accurately as the lines on your measuring tool. So you know what's, you can go from one line to another line, and then you kind of guess in between those two lines, and the marking sizes. And so the smaller the marking sizes, the more precise your guess, or the closer to the real value your guess will be. So you can get a better accuracy, and if you have, or I guess you get better precision if you have the smaller divisions. And so you want to use the one with the smallest divisions, where you can still measure out what you need to measure out. Okay. You can also, so with the micropipettes, these ones I showed you before were the single channel, which is the ones we most commonly use. But there are also multi-channels and repeater pipettes, which are when you need to basically do a lot of pipetting at once. For the multi-channel, instead of having just like a single tip, you have a strip of tips. And so typically these are going to be like 8 long or 12 long, so compatible with your typical PCR strip or plates or things that we'll talk about. And basically these multi-channels are then going to allow you to pull up the same volume in a number of different tips and dispense it. Now what you can do is, if you're pipetting the same thing into all those wells, they have these reagent reservoirs, where they basically these little plastic boats that you can stick your liquid into, and then you can suck things up. The downside is you're going to have more liquid than you would actually need to pipette, because it kind of gets spread out. But you don't have to have things in individual tubes. If you have different things you're wanting to pipette in each thing, or if you don't have enough of the liquid for it to be spread out as much, what you can do is you can actually be transferring from strip tubes or from a block directly into something else. So that can be helpful. So that's the multi-channel. You have the same volume in different tips. And with the repeater pipette, basically this is going to do the same volume, but it's going to use a single pipette. It's just going to do it over and over and over again. So you suck up the liquid once, and then each time you push down, you're going to get an equal amount pushed out. And again, you're going to have this in different size tips. This is really useful for making aliquots of things, and for like preparing gel samples when you have a lot, for preparing like stock solution aliquots that you're going to freeze down, various things like this, where you want to do the same proportion of the same thing over and over and over again. But if you have even more that you need to pipette, such as if you're doing crystallography and you're doing a bunch of screening, there are like robots that can handle liquid. This is an example called a mosquito. It does these like teeny tiny volumes for crystallography screens. We have liquidators, which basically there's automated versions and manual versions, where it will take a whole block at once worth and transfer it. So these are going to be really helpful if you have a lot of samples to transfer. Some other things that we can use to transfer liquid while measuring, syringes. So basically we have typically the syringes we use are Lurlock. So they have this kind of generic screw type base that you can then twist into a needle. And we have metal needles, which are like, you reuse them. And then we have like the plastic, or I guess they're both metal, but we have reusable needles that we use for things like when we're injecting into an ACTA or some other machine. And then we have the disposable needles, which are like the typical needles that you would see in a medical setting. they all have these adapters that are going to allow them to screw into the bases of these little lock syringes. And then these syringes, once again, are going to come in a variety of different sizes. In terms of, so these ones, they have the removable needles. Some of them have built-in needles, like these Hamilton syringes that we use often when we're like injecting a protein sample into an acta, which I'll talk about later. But basically, these typically have the needles already like attached to them. When you have the needles that you're going to be reusing, you want to make sure that you wash them really well. So like squirt water through them. If things get clogged, they have like little metal wires you can use to help declog them, but try to prevent them by getting clogged in the beginning by being sure to wash them right after you use them. And so typically what I often do is do like water and then do an ethanol wash. So those were syringes. And I should also mention that if you have, before we talked about the multi-channel and the repeater pipette, so all of this was things where we're actually measuring as we're transferring, but sometimes you don't really need to measure, you just need to transfer things from one place to another. And for this, we have a transfer pipette and a pass-through pipette. So a transfer pipette, basically these are just these little plastic things. They have a bulb at one end. They have little measuring marks, not very accurate, not very precise or that sort of thing. You just kind of suck up a little bit of liquid and stick it from one place to another place. And then they have kind of glass versions, which are pass-through pipettes, where you actually put like a bulb on the end. These are going to be really helpful when you're just doing things like trying to balance out a couple of tubes, maybe for the centrifuge, or you're trying to do some pH balancing. And so you're adding little drops of acid or base, trying not to overshoot. Okay, so that was just when we were measuring and we were transferring. What if we are just storing things? So some of the other most common equipment you see in a biochemistry lab when it comes to these like consumables is going to be your kind of like technical Tupperware. So like tubes and dishes and things. The most common tube we probably use is this Eppendorf tube. These are aka microcentrifuge tube. Eppendorf is the brand name, but it's kind of like Kleenex, and that name's kind of become generic for it. So typically these are about like 1.8 milliliters. And you use them in a microcentrifuge, which we'll talk about, or just to store small volumes of liquid. There are also these like cryo vials or cryo tubes. These are like Eppendorf size, but they are cryogenically safe. So basically you can stick them in your minus 80-degree freezer, which we'll talk about later, or in a liquid nitrogen or like LN2 storage. And so we commonly use these for storing like cell stocks for like tissue culture stuff. If you have a bigger volume, we have these like conical tubes, which we typically refer to as Falcon tubes. Again, another brand name. The most common sizes you'll see are like a 15 mil and a 50 mil. Now, these have a conical bottom, which makes them, and so the Eppendorf tubes, these make them really nice for when you're doing like centrifuging, that everything's going to kind of get pulled down to this bottom and just make a pellet here and things like this instead of on the side of the tube. Bad downside is that they're not going to stand up, and so then we have a bunch of tube racks. Just a little helpful tip is that, so for these PCR tubes, these are kind of like Eppendorf tubes, but they're smaller. Typically, these are about like 0.2 milliliters, I think, so like 200 microliters. And they're going to be, we can use them in a PCR machine, or we can use them if we just have small volumes. And I'll get more into PCR later. But you can, for a rack for these, what you can do is you can tape together two of the pipette tip racks, like empty rack things. That makes a good makeshift holder. And then they sell like plastic holders for these. These racks that I discovered recently are these like cool racks where they're metal and you actually stick them like in the freezer, or you just stick them on ice and they get really cold really quickly and they keep things cold and they help you keep your samples upright and stuff. Unfortunately, they're pretty expensive, but if your lab has the money for them, these are really, really helpful, especially if you have sensitive samples that you don't want moving around a lot. If you need your sample to float, such as in a water bath, which we'll talk about, they have these like flotation devices. You can often get these as swag from various expos, and it's when product rep salesmen come and things like that. These plastic ones too are really useful for if you're putting things in like a water bath or sonicator, or even an ice to hold tubes upright. Another tube which looks kind of similar to a Falcon, but it has like this rounded bottom, and instead of having one of these screw-on caps, it has one of these like kind of snap-on caps that's looser. This is a culture tube that you can use for like growing bacteria cultures, and these typically come in like 14 ml sizes. Speaking of growing bacteria, we have these Petri dishes, which we can use to grow bacteria in these like agar plates, which are kind of like bacterial pancakes. So basically you have this gel filled with bacteria food. And often what we do is we also add an antibiotic, and we can use this to select for cells that contain resistance to that antibiotic. And commonly we do this, we can use antibiotic resistance as a sort of selection marker by when we're doing cloning, and we can stick our gene of interest into like a plasmid, and then also stick on that plasmid will have an antibiotic resistance gene. So only cells that have taken in the plasmid will be allowed to grow, and then you see them grow as these like clumps called colonies. Much more on that in other posts. But that's the Petri dish, and then we can make the agar plates from them. And then we have some less like technical sounding things, like we sandwich Tupperware we can use for staining gels and things like this. Okay, we also have like blocks. So these are like deep well blocks. And then there's also these things called cluster tubes, which I find really helpful, where basically they're kind of like blocks, except that they have these removable wells, kind of. So these are almost like PCR strips, but they're larger volumes. And this can be really helpful. Okay, let's talk about some glassware. So there's a bunch of glassware with different shapes, different sizes. So basically all these different types of glassware are going to come in a variety of sizes. What really differentiates them is going to be more like their shapes and what we use them for. So beakers, these are basically, they look like your typical tumbler glass, except they have some markings on the side. And basically with the markings for the beakers and for the bottles and for the Erlenmeyers, these are all going to be approximate measurements. So you're not going to want to count on these measurements being accurate. So you don't use them for actually measuring, but you can estimate the volume based on that. And this is helpful for when you're saying, making a solution, you can kind of estimate how much water you're going to add, you need to dissolve your thing in, and dissolve it in one of these beakers before then transferring it to one of these graduated cylinders, which are going to be these long skinny things with the more accurate markings. And these are going to be, so you can dissolve it in a beaker and then pour it into a graduated cylinder to actually do the measuring. But you want to be careful because the lines on these beakers are not going to be very accurate. And so you want to make sure that you're not going to overshoot it with how much you add, but it gives you a rough ballpark figure. There are also measurement lines on the bottles. And again, these are just approximate volumes. One more thing with the bottles, so we typically these are like Pyrex bottles. So Pyrex is basically like the type of metal that's really sturdy. With these bottles, we're often sticking them in the autoclave a lot, which is this like heat sterilizer. And the bottle caps can kind of get all cracked, like old and crumbly. So you want to make sure that the cap you're using is not crumbly, or else the crumbly stuff can fall into your solution, which is not good. The cap should also have like an impact tack, or the lid should also have like a plastic ring around the top to help keep things sealed. And these bottles typically have this standard kind of top size that's going to make them compatible with various bottle top things like filters. So the beaker is going to be kind of like your tumbler, like your typical just cylinder. The Erlenmeyer is going to be more like pyramidal, and then you have your graduated cylinders are going to be these long and skinny. In terms of the measurements, so the graduated cylinders, these are going to be more accurate than the measuring is going to be more accurate than the beakers or the flasks. Again, similarly to how we saw with the pipettes, you're going to want to use the smallest thing that can do the job. Because your measurement is only going to be as good as in between the lines that you can see, and the closer together those lines are, the closer you can get to the volume that you actually want. And these are going to be calibrated better so that the volumes on the sides are actually going to be more accurate in terms of representing the actual volume that's inside. And so, yeah, when you're measuring one of these, you're going to want to make sure you're measuring from the meniscus. So basically the bottom of the meniscus. So where you have that little smile, basically the water is going to be pulling up on the sides at the surface. The water kind of gets attracted to the surface of the sides of the container, and it kind of pulls them up. This is going to lead this to the smiling effect, and you're going to want to measure from the bottom of the smile. And then because you're kind of having to, you can see which line is between, which lines is between, and then you kind of guess which it's in between. So the closer together those lines are, the more precise you can be. And then the more accurate these lines are in terms of what, like saying that this line really does represent whatever volume it says it represents, like the accuracy. Those are going to be more accurate for these graduate cylinders than for like the beakers and the flasks. So in addition to having more lines, so you get better precision, they're going to be more accurate. But if you want really good accuracy, what you're going to want to go to is often the volumetric flask. Now, I don't use these much, but you use them a lot in like chemistry and things. They are useful for if you need to have a really, really exact volume, but they're not useful if you need to have different volumes. Volumetric flasks are good if you have a single volume that you want to measure out a lot, and you need that to be really, really accurate. But you can only measure a single volume with a single flask, meaning that you have like a 1,000 milliliter volumetric flask, and you'll have a 500 ml, and you'll have a 250, et cetera, et cetera. But each of these can only be used for that single volume. Because how these volumetric flasks work is they have at the bottom, they have a kind of like bulb, and then they have this long skinny stem. And on that stem, they're going to have a line, which is the measure mark. And that measure mark is going to be calibrated. So it's really, really, really precise and accurate. And it's going to basically, you can know that if you measure so that the bottom of the meniscus is on top of that line, that's going to tell you it's exactly that volume. But it only has a single line. It's only basically calibrated for that one volume. So if you want to use another volume, you have to use another flask, and you need to have a flask that actually has that volume. So you typically are restricted to using fairly standard volume sizes when you're doing these. Okay, so that was our glassware and our pipettes and things. What else? Well, you incubate. So incubate is basically just a fancy word for when we stick something at like under constant conditions. So often we're talking about incubating at a constant temperature. We can also be incubating with constant CO2 and humidity, such as when we're dealing with incubating mammalian cells, where they need to have controlled CO2 and humidity. Some incubators are going to be shaker incubators, which we often use for cell culture for like bacteria or for other cells that we're growing in suspension. And then we have different sizes. So you have ones that can just do little tubes. You have ones that can hold a variety of different rack sizes, up to big shakers for big flasks when we're doing like protein expression and things like this. When we're growing suspension cells, we typically do it in one of the shaker incubators, so you can keep the cells from settling. When we're growing adherent cells, so basically where they're stuck to the surface of the plate, they're going to basically be in these, often they'll be in these sorts of Petri dishes, but these are typically cell culture treated. So they have a coating on the bottom that's going to make it so that the cells stick better. So you often don't use the same Petri dishes that you use for an agarose plate, but they look really similar. Also, when you're using adherent cells, you might see like flasks and plates. So these are dishes. These are like 10 centimeter dishes, which is your typical size. You also see like 15 centimeter dishes. This is referring to like the diameter. So the 15 centimeters are going to be bigger. You also see flasks, which are they're for adherent cells, but they look more like flasks, but they're flat and you lie them on their side. These are typically named in terms of their surface area. So you'd have like a T75 would be a 75 square centimeter, a T225 would be 225, et cetera, et cetera. Much more on cell culture and other posts, but basically those would be your flasks. Then you have your dishes, and then you have your plates where they're divided into numbers of different wells. So that's all for cell culture. What else you might see in a cell culture lab? This is a hemocytometer, which is used for counting the density of the cells. when you're trying to figure out if they're too crowded and you need to split them or break them up and give them more food. And so you can do this manually. Or nowadays there's electronic cell counters such as this Countess, where you put your sample in a little plastic slide and then you stick it into a machine and it does the measurement for you. So that can be really helpful. But warning, I've seen a lot of inconsistency between the measurements, so you might want to take multiple measurements and kind of average them. In terms of heating things, there's a lot of different ways we can heat things in the lab. So these incubators typically have a heating apparatus. So these are one way you could heat things. Other ways, if you want things really hot, these things aren't going to get super duper hot. They're typically made so that they'll be working at 37 or so, which is bodily temperatures and things like this. But if you want things really hot, you can microwave. We actually do microwave. Not very precise, of course, but this is really good for if you're trying to make agarose gels and you need to get the agarose to dissolve. When you're trying to destain gels, things like this, the microwave comes in handy. In order to get a flame, such as if we're just trying to keep a sterile environment, we can use a Bunsen burner. And so basically you have this little flint striker. This is hooked up to a gas line. You turn that on and you strike and you get this flame. For heating tubes and things like this, we have water baths and bead baths. A bead bath is basically kind of like a water bath, except instead of using water, it uses these kind of metal beads. This is going to be easier to sterilize and things like this. So you often see these in cell culture rooms. Whereas the water bath is going to be kind of this is a fairly standard thing that you'll find in most labs. Don't confuse the water bath with the bath sonicator. So the water bath is just going to keep things warm, but the sonicator, what it actually does is you use it for ultrasonication, where basically you send out these ultrasonic waves through the liquid, and then they're going to kind of cause these bubbles to burst, which is going to help do things like break up clumps and break open cells and things like this. So really helpful. And they have these bath sonicators where you stick things in this water and then the signals coming through the water. They also have probe sonicators where you basically have a metal probe and the signal's going to be, those ultrasonic waves are going to be sent from the probe and you stick the probe into your sample. But that was getting off track from our heating apparatuses. And you can set different temperatures. And these are good if you have something that's not a set tube size, because these can hold weird size bottles and things like this, such as your tissue culture media. If you have more standard tube sizes, we have sand blocks and heat blocks, where basically they have these metal blocks and they often have sand in the bottom, and you stick your tubes in there. These, at least the older ones, it can be hard to set an exact temperature. You often have a thermometer sticking out that you're trying to then twiddle the knob to get it to the temperature that you want. They also make electronic versions, which are a lot easier to use and I trust more. Often what you see is these also have built-in shakers. This is a thermal shaker. Don't confuse this with a thermal cycler. So a thermal cycler is a PCR machine. And so PCR is this technique that we can use to make lots of copies of DNA in little test tubes, those teeny tiny test tubes that I was showing you before, those PCR tubes. But yeah, so the thermal shaker is going to be basically your heating block, but it can also shake, and you can set the temperature more precisely. And you can also, sometimes you can they have ones where you can set different programs. Some of them, they actually cool down and things like this as well. Sometimes the thermal shakers have a cooling, but they don't always. The thermal cyclers, they pretty much always have both heating and cooling, because in PCR, you need to get really hot and then cool really quickly over and over and over again. So the thermal cycler is going to get hot and cold a lot faster than the thermal shaker, but it's not going to shake, and it's going to be restricted to using PCR strip tubes. Whereas with the thermal shaker, sometimes they have one set tube size, such as often an Eppendorf tube size. Other times, and sometimes they also have a mini Eppendorf, so a 0.5 mil. Other times these ones, this has a removable block where you can interchange it. So you can stick a deep well dish or something like that on. So it all depends on the version that your lab has. But the thermal shaker, this is going to shake. We use this for like incubating and shaking and things. The thermal cycler, the PCR machine we use for PCR. We also use for other things, including incubating when we have strip tubes. It can be really helpful if you're preparing a lot of gel samples, you need to boil them and things like that. So that was some ways that we can get things hot, but often we want to get things cold. And we have a variety of rays. We have lots of fridges and freezers of various sizes, and some of these are like under the bench. Some of these are going to be your typical like kitchen freezer, kitchen fridge type situations. Might be out in a hallway, might be shared in like a common area. We also have, sometimes we have deli fridges. These are commonly used for housing our protein purification equipment. I'll talk more about later, this like FPLC or ACTA. Cold rooms, which is my least favorite place, where it's like a walk-in refrigerator with benches and things like this, so you can actually do work in there. Often when we're doing work with proteins that need to be kept, that are sensitive and we don't want to hurt them. If we want to get things really cold, we have this -80. So often what we refer to like this normal freezer as like a -20, and a -80 is this really, really cold freezer. And you want to, when you're working with these, keep the doors closed as much as possible, or then you're going to heat this up. You're going to get these BP alarms going like crazy, and you don't want that to happen. Not to mention that it's not good for your samples. So -80, this is going to be these super duper freezers, and then -20, it's your normal freezer. Talking about like 4 degrees Celsius, that's going to be your typical fridge. If something says to store it at like 20 degrees or whatever, that's just going to be room temperature. If we want to get things really cold, really fast, we have liquid nitrogen. So this is often held in like these liquid nitrogen tanks, where they have this knob where you can turn it to let the liquid nitrogen out of here, typically into some sort of transfer dewar. A dewar is kind of like a thermos type thing that you can use in order to store this liquid nitrogen and not have it just dissolve off or evaporate off. We also have, so we have transfer dewars, which you can use for just like transferring the nitrogen from one place to another, as well as storage dewars for like long time storage. So we often use these dewars for storing like cell culture stocks in boxes, and like these big racks on these things that pull out, and you have these racks of tubes and boxes and things like this inside of one of these. Okay, so yeah, we also have these little like foam devices that we can use to store liquid nitrogen and flash freeze samples in and things like this. You're also going to see a lot of just normal ice buckets, as well as styrofoam. It also often does the trick. And again, when I showed you earlier, that's kind of like the next to the flotation devices were those plastic things that can be really helpful for holding your tubes in place, as well as those cool racks for when you need to keep the samples cold on the ice. Speaking of ice, there's a couple of different types of ice. So your typical ice is going to be wet ice, and then you also have dry ice, which is solid CO2, and this is going to be colder than the wet ice and drier. We often use this if we need to get things really, really cold, if we need to get things cold really fast, not quite as fast as liquid nitrogen, or when we're doing like shipping across the country or something like this, the dry ice can last and keep it cold for a long time. Let's talk about shaking. So there's a lot of ways where we're going to want to shake, times and ways where we're going to shake a sample or stir a sample to get things mixed together. The orbital shaker, this is going to be kind of like it gently goes in these orbits. And these are really good for like incubating gel membranes when you're doing a Western blot, or destaining and staining gels, basically where you just want to help, you have some sort of liquid and you want to kind of just swirl it around a container, help out diffusion, help like, so if you were to stick like a dye molecule into a thing of water, you would see the dye molecule spread out through diffusion. Basically, it's like random movement that leads to the overall spreading out into evenly. And you can help that along with help keep things moving with orbital shakers. So this is kind of just like going in these flat circles. If you want things to go kind of actually like be rolling, we have these hot dog rollers, as well as these end over end rotators. Also have like platform rockers where this is basically going to rock from side to side, again, often for like gels. And then there are nutators, which are kind of going to go up and down and circle at the same time. So those are different types of things. So you might use these for, say, like mixing solutions. This is going to be better for mixing solutions, whereas these guys are better for if you're trying to like stain or destain a gel or incubate a membrane with a wet in one of these boxes on the Western, where you're using the boxes, then you're not going to want to go like sending end over end and then have everything spill out. But they're like more gentle and stuff too. Okay, so that was just kind of like shaking and stirring and things like that. Oh, speaking of stirring, often what we're doing, so we talked about the shaker incubators, often what we're doing when we're stirring is we're actually using a magnetic stir bar in one of these heat plates. So I didn't even mention a hot plate before when I was talking about heating things. But we have this hot plates, and often what we're using is these also have a stir function. And so sometimes you have one of these magnetic stir plates that has heating, and sometimes these magnetic stir plates don't have heating. So make sure if you want something to be hot, you're actually using one that has heating. And then make sure if you don't want something to be hot, you're not accidentally knocking into the heat function and turning the heat on. But you stick a magnetic stir bar in there, and then this is going to stir while your sample is heating, or if you don't turn the heat on, not while it's heating. But this is going to be really good for when you're trying to dissolve solids. And then, so remember, when you're dissolving solids, you typically do it first in like a beaker, and then you pour it into a graduated cylinder. Make sure you don't pour in the measure drop bar. So I often use it, like hold a magnet on the outside to make sure the magnet, that next stir bar doesn't fall in. And then you can adjust the volume in that graduated cylinder, but don't trust the volumes on that beaker. That was getting a little off track. We were still talking about stirring and shaking and stuff. Speaking of like shaking, this vortex, this is going to basically shake things really fast. And that can be helpful for various things where you want to kind of break up clumps or just shake things really fast for some reason. If we're talking about like actually like spinning down the samples, so we want to centrifuge something, we're going to spin them really, really fast. Then we're going to want to use a centrifuge. And there's a bunch of different types of centrifuges depending on the speed you want to centrifuge things at, and the size of the things that you want to centrifuge, whether you need to keep things refrigerated or not, whether you need spinning bucket or hanging buckets, which I'll talk about in a second. But you have your basic, if you just want something like a quick spin with a small tube, like an Eppendorf tube, we have these like pulse microcentrifuges, as well as then we have these benchtop microcentrifuges. Again, these are used for like these Eppendorf tubes. And then we have a variety of centrifuges that are either like benchtop or floor model, where they're actually on the floor. And then we have the ultra centrifuges, which are going to go super duper fast. Centrifuges can be like fixed angle, in which case the tube is held at like a single angle. And they can be swinging bucket, where the tubes are, where the racks are kind of like the tubes and things are kind of in these buckets that can then swing. Much more in centrifuges and other posts. But these things, these are the rotors, which is what the tubes go into. And so some centrifuges have like interchangeable rotors where you can change out if you want to say have different size tubes, or you want to have plates instead of tubes. And so you have these swinging buckets and the fixed angle. And then those can be both in normal centrifuges as well as in ultra centrifuges. So ultra centrifuges, these go super duper fast. And again, you have fixed and swinging buckets. And we often keep the centrifuge rotors in like a refrigerator, and much more in centrifuges and other posts. But the ultra centrifuges is going to go really fast. This is good if you're trying to do some sort of fractionation, or you're doing some sort of gradient separation, something where you need to spin things really, really fast. So that's the ultra centrifuge. Okay, so let's talk about measuring things. So I mentioned before about some of the basic ideas about how you want to have something that's going to give you the most precision, so kind of give you the most digits you can get, as well as the most accuracy. So make sure that the digits it gives you are really representative of what's actually in the sample. When it comes to weighing things, we're going to have a couple different options. Typically, you have this like kitchen scale and this analytical balance. And the scale is going to give you fewer digits. So if you were to weigh the same sample on this analytical scale and a kitchen analytical balance and a kitchen scale, you can see that this would go to like 1.00 grams, whereas this would go to 0.9999 milligrams. So you're getting more digits. And it's going to be also be more accurate. So that's going to better represent the true weight of it. Things can get complicated when we're talking about accuracy versus precision with these scales and balances and things. So I don't want to get too complicated into it, but know that if you need your sample to be really accurate and really precise, you're going to want to use something like an analytical balance, but you can't use it for as big of heavy of things. With these analytical balances, they typically have these like sliding doors so that they don't even get disrupted by air. So much more on accuracy and precision and significant figures and things in another post. But just remember that you only know as much as your measuring tool knows. Next to the balances, there's often a pH probe. And so basically this is this probe that you stick into your liquid, often on a stir plate, so that you can be stirring while you're measuring. You stick this probe in here and it will measure the pH for you. You often have to calibrate it first with some known standards. And then you can dropwise add, using one of those transfer pipettes, add like acid or base to try to get the pH to the value that you want. Speaking of measuring things, we can also measure like light absorption. So there's like a spectrophotometer. These are more of like your classic tools for measuring light absorption. You have these little cuvettes, these little plastic like rectangle type things where you have your sample in here, and then the spectrophotometer is going to send light through, and the light's going to get absorbed, and the more the stuff that's in the tube, the more of the light that's going to get absorbed. And so you can measure the absorption to figure out how much stuff is in the tube, and that was a much simplified version. You can also do things at different wavelengths that'll tell you different things. I'm not going to go into it in this post, but that's the spectrophotometer. And so typically see you here you're dealing with these larger volumes. They have ones where they have smaller volumes in here, or you can dilute your sample so you don't have to have a whole like mill or whatever sample. But if you have a really precious sample, you don't have that much, or if you're feeling lazy, we have this NanoDrop, where basically you just stick a little drop of your sample, like a microliter to 2 microliters, onto this little metal dot on here. And what it does is this part comes down, and it creates this tiny mini column. And then that mini column of liquid is like what's happening in this cuvette, and the light's going to be shown through that mini column of liquid. And so this is an older NanoDrop, a newer NanoDrop. You can use these for measuring the concentration of DNA or protein typically. Okay, so moving on from measuring to actually more like doing stuff. Sometimes it's a good thing to suck. So we have these vacuum lines. You'll see a lot of vacuum lines in the laboratory. You also might see next to them like a gas line. Be sure you don't confuse these. If you turn on the vacuum and you're wondering, or you think you turn on the vacuum and you're wondering why things aren't vacuuming, you start to smell a gas smell, you probably did the wrong switch. And so the vacuum switch is going to be the one hooked up to the vacuum line. That then you can hook it up to various things. You can hook it up to like a vacuum filter if you want to filter a solution. You can hook it up. This is a vacuum manifold apparatus type thing that you can use for, sometimes we do a lot of like spin column purifications. Sometimes these columns, you can do some of the steps actually on a vacuum instead of spinning to centrifuge them. So this is one of these like vacuum manifolds, which can come in really handy, especially if you have a lot of samples you're dealing with at once, so you don't want to have to keep taking the tubes in and out of the centrifuge. But the vacuum line, and so the vacuum line, you can hook it up if you want to like filter solutions. These are both like single-use disposable, or at least technically single-use. You can actually reuse these especially if you're using the same sample. And they have versions of them that are reusable, like that you wash and things like this. They're made of sturdier plastic. and interchangeable membranes. But basically these have a filter, and then use a vacuum to kind of pull through. They have this membrane, and you use the vacuum to suck the liquid through the membrane, purifying it in the process. And so this is used for like sterilization and things like this often. So those would be vacuum filters. Another way we could filter things is like a syringe filter. So going back to the syringes we talked about earlier, sometimes we don't really care about what measuring marks they have. We just care because those measuring marks aren't going to be super duper accurate anyway. We just care about using that one that can hold what we want. And then we can use it to push the liquid through a syringe top filter, a syringe filter into different tubes. And note that these filters are going to come in a variety of different sizes, if you want things to be super duper pure and sterile, you're going to want to use like a 0.2 micron, a 0.22 micron filter. If you don't care as much about sterility, but you just want to make sure you get big clumps out, and maybe your sample is thicker, so you don't want to have to try to push it through tiny holes of a 0.22 micron membrane, but it's basically telling you about how tiny the pores are. Then like a 0.45 is often used, like even like a 1.2 or so if you're trying to deal with some cell lysate. These also have different like ring sizes, and so you want to, the smaller this ring part is, the smaller the surface is, the harder it's going to be to push through, but the less you're going to lose being absorbed on here. So if you have a larger volume, you can get away with losing some being absorbed into the ring. So the bigger ring size would be helpful. But if you have a smaller amount, you're going to want to use a smaller ring, but you might have to push a little harder. Okay, so those are syringe filters and vacuum filters. Those are ways we can sterilize, especially if we use that 0.2 micron. Another way we can sterilize things is an autoclave, which is basically a super duper hot oven sauna type thing. These often we have these tape on the top, this autoclave indicator tape, and it starts one color, and then when you put in the autoclave, it's going to like turn black. So they have like lines or words on them. I really want to make ones with jokes on them. So far, no one has taken me up on that offer or that suggestion. Not that I've like seriously offered it or anything or suggested it, but I think that'd be cool. Basically, it's going to change temperature when it gets hot, so you know that the autoclave was run on them. Typically, we put our samples into the autoclave in one of these in these plastic racks. We have these big Elmo gloves because that thing gets really, really hot. And yeah, we often use the autoclave when we're preparing solutions for things like working with bacterial cultures, so bacterial food, like the agar for those agar plates we were talking about before. Going back to filters, these are technically filters, but often we think about them more in terms of concentration. These like spin concentrators are these ultra centrifugation devices. Basically, they have this membrane inside of them that's going to have a different pore sizes. You choose a pore size based on the thing that you're trying to concentrate. And so often using this to concentrate proteins. So you want to choose a size that is smaller than the protein that you want to keep. And then your protein is not going to be able to go through the membrane, whereas the smaller stuff is going to go through the membrane. Your protein is going to be retained up here. Because you're pulling out all the smaller stuff, you're going to have, and the liquid and things, you're going to have your protein become more concentrated. Much more on these in another post. Key thing is that you're going to want to choose a size that's going to be like at least half as, like the size is going to be more than half smaller than the thing you want to keep. So if you had a like 100 kilodalton protein, you would want to use, say, a 30 kilodalton cutoff rather than, say, 100 kilodalton cutoff, because you want to make sure that these are just approximate sizes, and you want to make sure that your protein is not actually going to go through. But the smaller the pore size, the longer it's going to take. And then you stick these in the centrifuge, and it's going to that's going to help pull things through. Again, much more on these in another post, but if you see something that looks like this is probably going to be a centrifugal filter. Although they also have versions of these that are just kind of like filters. They just have a filter membrane that's going to have bigger pore sizes and not be for concentrating, but just for filtering, just like we saw with the syringe filters and things like that. So be careful that you don't get those mixed up. Now for a few things that are more familiar to you, probably things you find around the house and that sort of thing, maybe just some different versions. We actually have a lot of different versions of water in the lab. depending on how pure we need that water to be. So we typically don't use normal tap water for much of anything other than just like washing dishes. And even for that, we only use it basically for the initial steps of the washing. If you look at a sink in a biochemistry lab, you often see that there's a couple of different spouts. The one on the right is typically like your normal tap water, and the one on the left is going to be like deionized or DI water. So it has the salts and stuff taken out. Often what we'll do is we'll wash initially, which is like the normal water, and then rinse it with the DI water. We don't want any sorts of salts or any other things kind of drying on the surface of the flasks or that sort of thing, and then messing up the carefully calculated things we want. So when we're making a solution, we definitely not want to use the tap water unless it says to use tap water or something. Instead, we're going to want to use DI water, or even more commonly these days, we use this like Milli-Q water, which is the super purified water. Often labs have this like water purification system. It goes through a variety of filters and things to take out everything from the water except the water, and sterilizes it as well. Although when then you have it in the skewers, this can get kind of like, you can get stuff growing like in the tube, like in the spout and stuff here. So make sure you keep this really clean. And if you need water that's sterile, like go straight from the source of the Milli-Q and maybe even autoclave it. But anyway, you want to take all the other stuff out of the water, because when you're making a solution, you then want to have it so that that solution is going to be exactly what you tell it to be. And so you're going to add all the salts that you want, and you don't have to worry about whatever salts and things are already in the water. So we often use this Milli-Q water for things like making buffers as well as other things. And so actually when we're doing washing dishes, what we often do is I do like 3 washes with tap water, 3 washes with DI, and then 3 washes or like rinses with this Milli-Q water to get things really, really clean and nice. That's not, yeah, and then sometimes if you want things sterile, then you also then stick it in the autoclave. So we can get things really, really clean. And make sure you clean as you go so that things don't get caked on and gross, and don't just leave your stuff in the sink where then you're builds up and smells and your lab mates get mad. If you need if you need a small amount of water that's really, really pure and sterile and things, they sell this like molecular biology grade water, basically. Super duper purified water. It's guaranteed to not have RNases and DNases and things like that, which is one of the biggest things that we're concerned about a lot of times in biochemistry. It's not just the salts and things like this. It's not even bacteria and things like that necessarily, but it's the proteins and things that they make, such as these RNases that they secrete to kind of protect themselves. But these RNases will then chew up RNA and can degrade your samples. And so this water is going to say like RNase free. Yeah, I think it says here somewhere. But anyway, yeah, basically these tiny little bottles of water. And you might even want to take aliquots of that so that you don't have to keep sticking your pipette in there. And then each time you stick your pipette in there, you could then be contaminating it. So you can make little like samples of little bottles of the water from the bottle of the water, like little tubes of the water that you can then work out of. Some more things that you might find around your house. We use foil for a lot of things. It's good for keeping light away from light sensitive things like fluorophores or fluorescent dyes. We have your salts. There's a lot more of it. Instead of like a salt shaker, we have these giant tubs of sodium chloride, your typical table salt. Saran wrap, this is really great for if you're trying to wrap up like homemade gels and you want to keep them moist. Funnels, microwaves we already looked at. Bleach for bleaching solutions to kill things. Fantastic. This spray is actually really good for cleaning up like radioactive spills, like small things. Water repellent like Rain-X. This is great for your gel plates. If you want to make sure that the gel doesn't stick to the plate, you can use Rain-X. So there's various uses that we find for common household things. Also, we use a lot of just like office supplies. Lab tape comes in a lot of different colors. It's super duper helpful if you can develop a sort of like color code. Okay, now some things that are less familiar that you probably wouldn't find in your house, although it would be kind of cool if you did. In biochemistry and molecular biology, you're probably going to be running gels. Now we can talk about gels as we can talk about nucleic acid gels or protein gels. In both of these cases, we have this gel mesh, and we're basically sending the molecules using electricity to send the molecules traveling through this mesh The molecules, the bigger they are, the more tangled up they are, and therefore they're going to travel slower. And when you turn off the electricity to stop their motion, the bigger things are going to be higher up in the gel. So for nucleic acids, if you're dealing with like a DNA gel, you're typically dealing with one of these horizontal slab gels. If you're dealing with a protein gel, you're typically dealing with one of these like SDS page gels. This is going to be in this horizontal apparatus. We also have horizontal or vertical apparatus. We can also have vertical apparatuses we can use for like page electrophoresis. So page is polyacrylamide gel electrophoresis. And we can use these gels also for separating like small DNA or RNA pieces because it's going to have a finer mesh, which you can get better separation. Much more on these in other posts, but in any of these cases, you're going to need some sort of power supply. And so you have like a power supply, and then you have your gel tank and your electrode system, which you then stick your gel into. You have the lid on the tank and things like this, and much more on this in other posts. But another thing you might see hanging out around there is going to be these like kind of like gel knives and gel cassette crackers and things like this, which you can use to actually break open the gel cassettes. So sometimes if you buy them pre-made, they're going to be plastic. If you make them yourself, they're going to be glass. But if you want to take the gel out from between the plates, these are going to allow you to do that. And then I really like these metal ones because they're good at then if you want to like cut the wells off or something like that. These plastic ones, the bottom can get kind of chewed off, and then when you try to go and actually cut your gel, it's going to tear. So these metal ones are really helpful. But anyway, that's just a technical note. This might look like something you find in your house, but it's way, way cooler. So parafoam, it's basically like this super thick cling wrap, saran wrap type stuff. And it's super duper sturdy. You can stretch it for oodles and oodles of length. You can use it for a ton of different things, such as like wrapping Petri dishes, sticking it over the top of graduated cylinders when you're trying to mix solutions. One of my favorite things is to use it to actually help you load gels. Basically, it's kind of waxy, so the water's just gonna kind of beat up on it. And so you can use it then in order to allow you to mix your sample with like the loading buffer and things like this. So parafoam has a lot of really cool uses. But going back to our gels, we're going to want to look at the gels. And so there are different ways we can look at gels. The standard ways, if you have like a dye gel where it's like a visual color dye, such as if you're doing something like a chromacy staining of your protein gel, you can use just like a light tray in order to get a better look. If you want to then take a picture, we have these like gel docs or chemi docs. They have these white trays that you can use just for taking pictures of like colored things, as well as they often have some sort of UV tray. They also have like separate UV trays that are separate from here. In the gel doc or the chemi doc, everything's kind of happening within the confinements of this machine, and it also can take a picture. Typically, this picture can save to a computer, or it'll print out in one of these kind of like Polaroid type looking things. I don't know what you call those, the thermal printers. Well, it's kind of like old school printout things. But basically these trays, the white trays, are going to be used for when you just want like just normal light and then to take a picture. Then they have these UV trays. Sometimes they also have like blue light trays, where basically these trays are going to be used if you're using some sort of fluorescence, things like this. If you have some sort of ethidium bromide or easy stain, cyber gold, any of these sorts of nucleic acid stains, it will be able to then show you them and detect them. If you're working on one of these machines, everything's kind of like safely hidden away inside of this machine box. If you're working with a UV tray that's just out, you're going to want to make sure you're wearing your protection. You have your goggles, maybe even like one of those face shields. You have these plastic shields above the box, things like this to protect yourself from basically getting a bad sunburn. And so, yeah, so when you also don't want to have your sample on the UV too much, because then the UV can actually harm the DNA or the RNA. So that was the UV. A couple last things. So the ACTA, the FPLC, if you're working in a lab that's going to be doing some protein purification, we have this ACTA machine, this FPLC, so ACTA is the brand name. But basically it's used for column chromatography, which is a technique where we can separate proteins based on different properties of the proteins by sending them traveling through columns filled with these little beads called resin. Those columns are going to interact differently with different proteins, and therefore you're going to be able to separate the proteins, and then you can separate them as they come off of the column at different rates. You're going to kind of go through and come out into this fraction collector, and then you can collect the purified protein. You can also do it old school with like gravity flow chromatography columns. So these are the chromatography columns for like an ACTA, and they're typically pre-packed, and you don't take them apart or anything. But then you have these gravity flow columns where you can basically, they're just these empty columns with this stopper at the bottom, and then you can basically put in whatever type of beads and things you want. Okay, and then you just like gravity flow it through instead of pushing it through with pumps or pumping it through. Also, just try to reuse whatever you can in the lab, and just be creative about how you can organize your stuff with basic like trash and stuff lying around. I call it like recycling. But anyway, there's a lot of stuff that goes on in the lab, and hopefully this helped you feel more familiar with it. And if this is your first experience in the lab, best of luck, and it's a lot of fun, and it can be kind of intimidating, but it gets a lot more comfortable, and so hopefully it'll soon feel like your second home.
Video này do mình, cương bên npslab.vn, tự chỉnh sửa phụ đề hoặc lòng tiếng bằng công cụ, thấy vậy thôi, chứ tốn công lắm á. Thấy hay thì nhớ bấm theo dõi kênh, cho mình thêm động lực làm mấy video phòng lab xịn sò nữa nha.